It has long been thought that geographical barriers hinder gene flow within a particular species and ultimately affect the divergence of a population. This divergence can have an effect on a particular population's behaviour, connectivity, and the transmission of pathogens, ultimately affecting the conservation of the species. It was previously proposed by Inu et al. Study that the Malagasy River limits gene flow due to the lack of shared haplotypes between the north and south banks. However, the authors of this paper aim to evaluate the Malagasy River and prove that it is not an absolute barrier to chimpanzee movement, and as a result, the chimpanzees either side of the river should be considered a single population. To investigate this, 11 highly qualified scientists collected and recorded results regarding the location of nests, fecal samples, movements, and vocalizations of several chimpanzees within the Greater Gome region and the Greater Mahal region. 16 fecal samples were extracted from the north and south of the river and subjected to mitochondrial DNA analysis to confirm their species origin. The D-loop region of the mitochondrial DNA genome was amplified, and as a result, 13 samples yielded 3 different mitochondrial DNA haplotypes, all of which had been previously identified to cluster with eastern chimpanzee sequences. All genotype fecal samples were subjected to Simian immunodeficiency virus detection and all screened for HIV. This procedure involved analysing the presence of antibodies and using a highly sensitive and specific Western blot approach. As seen in Figure 4, 2 chimpanzees exhibited clear evidence of Simian immunodeficiency and HIV infection. Rudisell et al.'s previous studies on SIV suggest that some mitochondrial DNA haplotypes from the south and the north correlate, but it was unknown whether this was due to recent gene flow or common distant ancestry. The results shown indicate that recent gene flow is in place, as the 2 infected subjects were found from opposite sides of the river, ultimately suggesting the locations are epidemiologically linked. Further evidence includes the discovery of a natural shallow ford, which was able to be crossed on foot by investigators. Whilst under examination, the 2 fecal samples, both obtaining the characteristic size, smell, and shape of chimpanzee species, were identified within the riverbed itself. This particular finding strongly suggests that chimpanzees are in fact able to cross the suspected barrier. The data gathered does not provide conclusive evidence of connectivity between the 2 regions, and thus the south and north chimpanzees cannot currently be considered a single population. Thank you.